It's nanotech. You like it?
A little something.
Mr. Stork, I'm being beamed up.
Give me a little juice, Friday.
You said save the wizard.
We're too high up. You're running out of air.
Yeah, that makes sense. Oh, come on! Oh my God! I should have stayed on.
Then we thought, well, could we make submersible nanomachines and drive them along? And I thought it'd be really good if we could do something like this. You know, I was hoping that these nanomachines at this level, if we could make them so that they'd be yellow, we could call it yellow nano submarines. And you guys don't know what yellow submarine is, but okay, you do. Okay, all right, okay. So I thought it'd be a great title, Yellow Nano Submarine, but they weren't yellow. And then I tried to get the Navy to get excited about this. And we could make 10 to the 18th submarines. So people had looked at molecules moving in solution, particularly not molecules, but nanoparticles. And they had they'd taken clusters where you had gold on one side, silicon on the other, and they'd do polymerization off the edges, and they'd try to move things along. Ayusman Sen at Penn State had done a lot of work on this sort of thing, and others have. And so we weren't the first to try to think of things moving in solution, but we were the first of trying to track single molecules moving in solution rather than nanoparticles, which are much bigger than a single molecule. So there's a lot of challenges in that. These nano submarines are going to be less than 10 nanometers in size. How do you track them? And what's the mode of propulsion? So again, working with Stefan Link's group, what you could do is you could take glass slides and you could put spacers on the glass slides, which are just polystyrene nanoparticles. So you just have a tiny little bit of solution between two glass slides, and the only distance between those two are nanoparticles that you can't even see these particles. And so you just have a very small volume there. And so you can have a very small volume, or you could have something just a few microns in size. So you can barely see these things, and then you have your liquid with your submarines in here. In this case, it'd be a water solution. Okay, so we had seen these before, but then I met this guy. So what happened was I had tried for a long time to take these nanomachines and have them drill into cells, drill right through the lipid bilayer of a cell to kill a cell. That's what I wanted to do. I wanted to kill cells with these nanomachines. We don't need the wheels or anything. We just take the motors and have them drill into cells and puncture through and kill them. This would be a great way to kill cancer, right? You have cancer cells and target it to a cancer cell. And I was asking around a lot of people, who were good at this. You had to know how to work with cells. You had to know how to work with imaging systems. And these were fluorescent enough that we could watch them using a fluorescent probe. Then I met Robert Powell, not at the rodeo. It's when he came to the U.S. that I took him to the rodeo here. But I was a lecturer at Durham University, invited to give a one-week lecture, and so I was giving a different seminar each day. And one day I was talking about the nanocars. And I had been asking one of the professors there, who I thought could really pull this off, hey, I'd like to drill into cells, we get a collaboration going, we'll make the molecules. And he said, at this stage in my career, I'm not sure I want to do it. It's a big job. He says, but I have a postdoc, Robert Powell, who's the best experimentalist I've ever had with me. And Robert had just gotten a job in the university as a lecturer A lecturer in the UK is sort of like an assistant professor in the United States. They start out at lecture. And so I talked to Robert for like, I don't know, 10 minutes or 15 minutes at the end of a seminar, saying I'd like to try to get these machines to do it. He said, okay, send me a sample, we'll work on it. I sent him the sample. When I got back to the US, I sent him some samples. And back in those days, this was like 2015, we didn't have to have all these forms filled out for mailing samples. Now we have, I mean, lawyers mess up everything. You got to have non-disclosure agreements, material transfers agreements, and all of this stuff. So anyway, I just sent them this, and like within 2.5 months, he had gotten it working. So the idea is that you put one of these molecules on a lipid bilayer, and then you turn these on, and they would spin, and they would drill through and pop holes in the lipid bilayer. And if you pop enough holes in a lipid bilayer, that cell is going to die because you lose your whole ionic potentials differences, so it's going to die. And you can actually see this. You look at it, here's a cell, and you see a time course here. You see these little bubbles on there? Can you see those? Can you see little bubbles on there? Yeah, that's called blebbing, and that means that cell has a hole in it, and the guts are just squirting out. That's what that means. So these cells end up undergoing necrosis. They don't undergo apoptosis. Apoptosis is a controlled cell death. It's controlled. It chops up the proteins. It chops up the chromatin, and then it slowly dies. Necrosis means that boom, they just explode, and they can't chop up their DNA. They can't chop up their proteins. They just die. And these would necrose that way. And so you can see this as a time course. So we just took these nanomachines. Look at these nanomachines. We took a control that doesn't rotate, and we had different end groups on here, and we used it in the presence of propydium iodide. So propydium iodide is a dye that turns red when it intercalates into DNA or RNA. Now, the only way it can intercalate into DNA or RNA is to get inside the cell, but it can't normally get inside the cell. So you put this in the medium with the cells. So this is just in a dish. It's not in a live organism. It's just cells in a dish. And you put propydium iodide in that dish, and it's not lighting up. But once it gets into the cell, boom, it'll intercalate in DNA or RNA and light up. So it's a probe to tell you when the cell is opened up, because there's propydium iodide, but it's by itself only gets in very slowly. And so in the blank, there's no propydium iodide going into this thing, and you shine a light on it for 15 seconds, 300 seconds, 450 seconds, 600 seconds. So then what happens, this is the time that it took to necrose. So the time that it took to necrose. And so what happens is, as you take this molecule 3, the time that it took to necrose, they started to necrose almost instantly, and the later ones, by the time they were done, it's here, starts here, ends here. And so it's happening much more quickly. And you can see the propydium iodide flooding this was just time to necrose. Here's the propydium iodide added, and you can see the propydium iodide just moves right on in, and it goes in and it even gets into the nucleus of these cells. So these are PC3 human prostate cancer cells. And so you can see that they get in, and then they allow propydium iodide to get in. And then we put these peptide addends on them, because you can look up in a book, you can say, okay, we're trying to kill PC3 cells. So it's okay, if you want a recognition for PC3 cells, PC3 will overexpress this glucose-regulated protein on its surface. So we're targeting this, and it's a 78,000 kilodalton glucose-regulated protein. So if it says, okay, this sequence of amino acids, this short peptide binds to this. And so we just order this peptide from a company, and then we coupled it through what's called a click coupling onto the ends of this nano machine, this rotator. And some of them we coupled on two ends, some we just coupled it on one end. And so now it should recognize it. So we're exposing it to 3 different types of cells: the PC3, the human prostate cancer cells, NIH3T3 cells, which is a breast cancer line, and Chinese hamster ovary cells. And but it should target just the PC3 cells because of this sequence here, and we could do that. So I don't have the slides here. I'll put them in for next time. But it would target just those particular cell types, and then you'd see them start going after those areas, and then we started killing the cancer cells, and that turned out to be a big deal, and we ended up starting a company around that to kill cancer cells and to kill superbacteria, which we'll talk about next time. But and then also treating cells. So you just pop a few small holes and you have drugs in there. So this opened up a whole new area. So we had set out to try to do construction with nano cars. That became very difficult because of the manipulation. But what turned out from this was to take the motors themselves and use it to go after killing cells in solution, rather than you don't need the wheels here anymore. Are there any questions? Well, this is nanotechnology at its finest. Okay, see you next time.